Welcome. In this lesson, we are in EOC review mode. So we're going to recap these first two standards talking about the relationship between the structure and function of cells and their organelles, but also standard 1.2, where we kind of analyze the cell as a living system. So dive into homeostasis and how cells are able to survive and how they're able to function. So starting off with our organelles, this is just a short list of kind of the most important organelles that you need to be familiar with for the EOC test. So this is by no means a complete list of all the organelles found within a cell, but they'll be the ones that you're going to be expected to know. So you've got your nucleus, your plasma membrane, aka cell membrane. Okay, so both words are kind of used interchangeably. Cell wall, mitochondria, vacuoles, chloroplasts, and ribosomes. So again, those are just kind of the big ones. So not only do we need to be able to identify the organelles as far as matching them with their correct function, we also need to be able to identify organelles, what they look like, so where they're found within a cell. So here we have a plant cell. We know it's a plant cell because of a couple of reasons. Number one, there's a cell wall. Number two, there is a large central vacuole. And then number three, we have a chloroplast. So those are the three organelles that you're going to find in plant cells that kind of give it away as a plant cell. We also have a nucleus, which tells us that this is a eukaryotic cell because it has a nucleus. Prokaryotic cells do not have a nucleus. So no nucleus. Plant cells also have a mitochondria for making ATP energy. They have that cell or plasma membrane, and then ribosomes. And these ribosomes we've pointed at are the little dots that are found here on the ER or on the endoplasmic reticulum. But ribosomes can also be found just kind of free floating out in the cytoplasm. So kind of going a little bit into the function of organelles, the mitochondria is one of the most important organelles, and the structure of the mitochondria gives away its function. So the structure determines its function. So for example, right, the folded inner membrane of the mitochondria increases the surface area that is available for the production of ATP energy during aerobic cellular respiration. So we can kind of see this like maze of like blue stuff in here. So all of that space is where aerobic cellular respiration happens. So the structure of the organelle determines its function. The organelles also interact with each other in order to carry out very specific functions, such as energy production and use, transportation of molecules, disposal of waste, and the synthesis of new molecules. So, for example, one of these interactions you have between DNA and ribosomes and mitochondria. So DNA codes for proteins, right, which are assembled by the ribosomes and then used as enzymes for the production of ATP energy within the mitochondria. So there you kind of have three organelles, the nucleus, the ribosomes, and the mitochondria, all kind of working together in almost like an assembly line in order to create ATP energy in the mitochondria. So a little bit on microscopes. You will need to be able to calculate total magnification, which is simply the eyepiece times the objective lens. There are three different objective lenses, and they'll tell you, high, low, medium. So the eyepiece is typically always going to be 10. And then let's say you have an objective, you're using the medium objective lens, which is 40. So your total magnification would be 10x times 40x, 10 times 40, which would give you a total magnification of 400. Okay. You do have two types of microscopes, electron microscopes and scanning microscopes. Electron microscopes are going to have a higher magnification than the light microscopes. I apologize, I said scanning, I meant light. So you have two types of microscopes, electron microscopes and light microscopes. And electron microscopes have a much, much higher magnification than light microscopes. Using an electron microscope, you can see, right, all of the individual organelles and things that are as small as viruses. Whereas a light microscope, you'll be able to see the cell itself, but not necessarily the little organelles inside of it. So prokaryotic cells, right, we talked about where you've got everything's pretty much divided into either being prokaryotic or eukaryotic. Prokaryotic cells are the original cells. They were the first to develop or evolve on Earth. They are much less complex compared to eukaryotic cells, so very small, very basic. They are always unicellular, and they lack membrane-bound organelles. So you will never find a nucleus, a chloroplast, or a mitochondria within a prokaryotic cell, ever. If you see a mitochondria, you're looking at a eukaryotic cell, so probably a plant or an animal cell. Okay? Bacteria are a really good example of prokaryotic cells. They are very simple, very low-key. So comparing, right, prokaryotic and eukaryotic cells, the presence of those membrane-bound organelles, right, again, is going to be eukaryotic cells. They are just not present in prokaryotes. Now, what you will find in both prokaryote and eukaryotic cells are ribosomes, because ribosomes make proteins, okay? You also have to have some type of genetic material, either DNA or RNA, okay? But again, the difference is that a prokaryotic cell, DNA is just hanging out in the cytoplasm, whereas in a eukaryotic cell, it's in a nucleus, okay? Prokaryotes are also going to have circular DNA, so they literally have like one chromosome. There you go. That is the singular chromosome of a prokaryotic cell, okay? Versus eukaryotic cells, right, we have that double helix, okay? And then again, prokaryotic cells, they're just much more, they're small, they're simple, they're basic, okay? So comparing and contrasting a prokaryotic and a plant cell and an animal cell, again, they are both eukaryotic cells because they have a nucleus. So we can see the nucleus right here and right here. Now, plant cells are going to have a chloroplast, they're going to have a cell wall, and they are going to have a large central vacuole. And typically, plant cells are going to be drawn in a box or a square or rectangular shape. Animal cells are typically going to be drawn with a circular shape, so circle shape. They do have vacuoles, but they are much, much smaller, okay? Animals don't necessarily need to store as much stuff as plant cells, because with animal cells, right, humans, we have animal cells, I can just get up and go get food. I don't necessarily have to store it. So our vacuoles are much, much smaller. And then certain animal cells also have flagella and cilia that can help with movement. So a good example of this would be a sperm cell. Sperm cells have flagella to help them to move, right, for sexual reproduction. All right, so cell differentiation. This is where we're talking about where all these different types of cells actually come from, okay? When you're talking about or thinking about multicellular organisms like humans, right, we have a lot of different types of cells. Lung cells, liver cells, heart cells, skin cells, right? All of these cells. And all of the cells, right, all of those cells begin as undifferentiated masses of cells. So an undifferentiated mass of cells is a stem cell. There we go. So a stem cell is where they're all the exact same. Well, that doesn't work for us, right? We're multicellular. I need skin, I need lungs. So through the process of differentiation, right, the variation in DNA and gene activity determines essentially the job of the cell and ultimately their specialization. So cells become specialized. So during the process of differentiation, very specific parts of the DNA are activated. Those parts of the DNA that are activated will then determine the function and therefore the specialized structure of a cell, because not all cells look the same. So again, structure determines function. Now, all cells in your body contain the exact same DNA, which is why DNA forensics even exists, like CSI. Every cell contains the same DNA. So every cell has the potential to become any type of cell. But once differentiation happens, it cannot be reversed. So once the signal is sent to that stem cell, hey, you're going to be a lung cell, that's it, game over. You're a lung cell. Okay? Nearly all the cells in multicellular organisms have exactly the same chromosomes and DNA. Again, forensics. If you know, my skin cell didn't have the same DNA as my hair cell, people would be able to get away with crimes. And then different parts of the genetic instructions are used in different types of cells. So, and again, that's influenced by the cell's environment and what the multicellular organism needs. So different genes are turned on, which differentiates a skin cell, right, from a liver cell, right? So that's cell differentiation. That's how multicellular organisms are able to survive and function. So these are a lot of the specialized cells that we have. Again, it's not all of them, but these are probably going to be the ones that you are going to be asked about on the EOC. So we have red blood cells, which carry oxygen, muscle cells, right, for movement, white blood cells for our immune system, and then nerve cells, right, to carry messages, right, nerve impulses. So you touch something, it's hot, you jerk your hand back. That's a nerve cell. But we can see, especially with the red blood cells, right, these cells, they're small, they're circular, right, which is a very different structure from this neuron over here. So structure determines function, okay? Now, specialized cells in plants, the two you need to be familiar with are the xylem and phloem. Think of xylem and phloem, right? They're vascular tissues. Think of them as the veins and arteries of plants. So you have veins and arteries that let you move oxygen and blood and nutrients, from one part of your body to another. Plants have the same thing. Xylem help to transport water. Phloem help to transport glucose or sugar, right? Food for the plant. So xylem and phloem for a plant is the same thing as veins and arteries for a human. All right, cell communication, right? Do cells need to communicate? Yes, absolutely, right? My brain cell needs to know what's happening with a lung cell, okay? So yes, cells communicate, okay? They communicate through chemical signals, right, that are released by one cell that can influence the development or the activity of another cell. Most of these chemical signals are going to be either hormones or neurotransmitters. Okay, those are the two types of chemical signals. But essentially, right, cells are communicating. So a signal is sent from one cell. The signal is picked up by a receptor on the secondary cell. The signal is transduced, or it's sent into the cell, and then you get some type of response. Okay, so that is your cell communication pathway. So signal, receptor, response. So stem cells, what we were talking about a minute ago, right, are unspecialized cells, right, that continually reproduce themselves, but under the appropriate conditions, i.e., right, differentiation, right, they have the ability to become any type of specialized cell. So stem cell, right, heart cell, lung cell, liver cell, nerve cell, whatever it is that the multicellular organism needs, right, that's what it's going to turn into, okay? You do have two different types of stem cells, which we'll touch on a little bit later in our biotech recap, because there's a lot of ethics around using stem cells. The most ethical concern is around embryonic stem cells. Okay, you find them in embryos, and then you have adult stem cells. they're found in adults. Okay, so you and I have adult stem cells. But scientists, right, they've demonstrated that both types of stem cells, embryonic and adult, under the correct laboratory conditions, can differentiate into specialized cells. This is really helpful because, again, as an adult, we need stem cells. Again, you break your arm, you need to fix it. So you'll take some of those adult stem cells and you'll turn them into bone, right, or skin because you've damaged yourself. So we need stem cells, right, for growth and healing and repair. And then embryonic stem cells, they're just more potent. They can differentiate into a wider variety of cells, but again, kind of controversial because they do come from embryos. But, you know, again, it's that bioethics, right? Where do we draw that line for what technology we're using? This would be a good place to pause if you wanted to take like a quick brain break, but we're gonna keep on going. So this is the last part of right, goal 1. We're gonna take a look at these last 3 standards, talking about homeostasis, talking about cell growth, and then also cell adaptations. So if you recall, homeostasis, right, is where you are maintaining a stable internal environment. And the way cells are able to do this is through the use of buffers. So a buffer is really any type of solution that can regulate pH. So I could add a buffer to an environment and make it, you know, more neutral. Okay. Homeostasis is maintained through temperature. So cells, right, they have to be kept at a certain temperature, a certain amount of glucose. So think about like your blood sugar, and then also water, right? You can't have too much or too little. So all of these are going back to homeostasis, maintaining that stable internal environment. So if the temperature gets too high, right, you have a fever, you're sick. Glucose levels, right, your blood sugar, it's too low, you pass out, right, you get dizzy. Same thing with water, right? Are you dehydrated? You know, to get some water. So all of these things need to be maintained, right, in equilibrium. So we've got to maintain homeostasis. Also talking about homeostasis is active and passive transport. So remember, cells are constantly moving materials in and out. Typically, materials that are coming in are going to be things like food, right, nutrients, things that are going to be moving out, waste, or other products that have been made that just need to be shipped somewhere else in the cell. Active transport is going to require energy, right, ATP energy from the mitochondria, because we are moving against the concentration gradient. So we're moving from an area of low concentration to high concentration. We draw my cute little picture. Right, so we've got our hill, there you go, and then here's, I don't know, someone. My drawing tool is quite terrible today. There you go. So, all right, here's your high concentration right up at the top of the hill, and then you can have a low concentration right at the bottom of the hill. So if I am moving right from low to high, essentially I'm moving against gravity. That's going to take a lot of energy. Okay, I have to bike up the hill. Half the time I can't even do it. I have to get off the bike and walk it up. Okay, so it takes energy, right, to go against the concentration gradient or to go against gravity. Passive transport does not require energy because substances are moving from an area of high concentration to low concentration. Okay, so again, you're on your bike and you're going down the hill. Much easier, right? Gravity does the work for you. It's the same thing in our cell. The concentration gradient does the work for us. So this is passive transport. It does not require any energy. So examples of passive transport are diffusion, osmosis, and facilitated diffusion. Remember, osmosis is specifically the movement of water. Diffusion is just everything else: oxygen, carbon dioxide, salt, whatever. And then active transport, you have endocytosis and you have exocytosis. Yeah, so again, osmosis, right, the movement of water across a membrane. So if you place a cell into a very salty or sugary solution, right, the water will move out because right, salt sucks. It's going to suck all the water out. So, right, because you're always trying to create equilibrium. You want a balance. So here in our picture down here, right, we have like a glass of water, right, and we have kind of a high concentration of, let's say, sugar, right, over in the top left. But over time, right, those sugar molecules have diffused, right, out through the water to be more even, right? So I wouldn't want to drink this first one because that would be pretty gross because I'd get just like a big gulp of sugar and then the rest of it would just be water, right? I want my lemonade to be more, diffused, okay? When it comes to osmosis, though, just remember salt sucks, okay? So the wherever your high concentration of salt is, it's gonna suck the water, okay? So that can cause the cell to either shrink or it can cause the cell to swell. It just depends on where that high concentration of water is. Talking about the plasma membrane a little bit more, remember this is another name for our cell membrane, plasma membrane, but it also goes by another name, which is the phospholipid bilayer. This is because the plasma membrane is made up of two layers of phospholipids. You also have a bunch of proteins that are embedded within the phospholipid, which allows for substances to enter and leave the cell. So we always say the cell membrane is essentially responsible for maintaining homeostasis. It is responsible for making sure that enough water gets into the cell, but also waste leaves the cell. So the cell membrane regulates homeostasis. So our cell cycle, which remember is responsible for growth and repair of somatic or body cells. So like you damage your skin, it'll go through the cell cycle to repair itself. So the cell cycle is a series of events in the life of a cell. There are two main phases of the cell cycle. The first one is interphase, which is all of the blue right here, okay? This is interphase. There you go. Interphase is made up of three parts. You have G1, or growth 1, S phase, and then G2. Okay. Interphase, you are preparing for cell division. Okay. So the second phase of the cell cycle is cell division, which is the other two slices of the pie. Okay. Cell division is made up of mitosis and cytokinesis. Mitosis is where the cell is actually dividing. So prophase, metaphase, anaphase, telophase. So I go from one cell to two. Cytokinesis is when you actually get that second one. Okay, so you need mitosis and cytokinesis together to go from one cell to two. Okay, but mitosis and cytokinesis, right, both together to make cell division. Cell division does not take very long. It takes like 60 seconds. So a cell spends a majority of its life in interphase, just kind of hanging out, doing its everyday thing, living its best life. But the minute it gets that signal to divide, it enters cell division. It goes through mitosis and cytokinesis. And again, mitosis, right, is used for growth and repair of somatic or body cells, okay? It is a form of asexual reproduction because you have one cell, right, that divides to make two identical daughter cells. Identical daughter cells. Whatever chromosomes you start with better be what you end with. So for humans, we have 46 chromosomes. You best have 46 at the end. If you've lost or gained any, you're in trouble. Okay, so whatever you start with, best be what you end with, right? Because it's asexual. They are identical daughter cells. And then the last couple things, talking about unicellular adaptations. So these are, again, specifically going to be unicellular or single-celled organisms. So the most famous ones that you're going to need to be familiar with are the amoeba, which is right here. The amoeba has two adaptations you need to be familiar with. The first one is a contractile vacuole. It is responsible for regulating water. A contractile vacuole is not the same thing as a regular vacuole. When we talk about a normal vacuole back in plant cells, that was just for storage. A contractile vacuole, right, it contracts. Think of it as like a bladder, okay? So it regulates the amount of water. Amoebas also have what's called a pseudopod, false foot. It's used for movement, okay? The other organism is the euglena. It has two special adaptations as well. It has a flagella, which is like a whip-like tail, again, used for movement, and it has an eye spot, which can help it to detect light because euglena are plant-like protists, meaning they have a chloroplast, so they can do photosynthesis. And then we have the paramecium. It has a specialized structure called cilia, which are these little tiny hairs that, again, are used for movement. Because it's very important for these unicellular organisms to be able to get up and go find food or avoid predators. They've got to be able to move. So those are kind of the main adaptations that you need to be familiar with when it comes to unicellular organisms. And then there's also a couple adaptive behaviors for unicellular organisms, most specifically chemotaxis, phototaxis, and geotaxis. Taxis just means movement, okay, like a taxi cab. So chemotaxis is movement because you're responding to a chemical. So a euglena moves because it senses that there's food nearby, okay? Phototaxis, right, responds to light, so photo, like photosynthesis, and then geotaxis is in response to gravity, geo meaning earth, okay? So that is our rundown, again, for this first topic, right, lesson kind of 1 on cells. So we've talked about a lot here. Let me know if you have any questions still, but that is it for this first set. Okay.